This UCSD TV program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest programs.
Good morning. It is a pleasure to welcome you again to this beautiful auditorium at the Scripps Research Institute for the second installment of the Science Saturday series. And today you'll have the pleasure of listening to one of my very esteemed colleagues, Dr. Ryan Shenvi, who will be telling you about very exciting ways to do science. And toward the end, also, if he gets a chance, tell you about his very exciting research with molecules. And so we wanted to thank today our sponsors, Gilead Sciences, who have kindly sponsored this series. And also I would like to thank Shirley King, who has been working very, very hard to get all of you here and put everything together, this wonderful program, and all of her staff as well as the UCSD TV crew who are here helping us with this program. And now I leave you with Dr. Ryan Shenvi.
All right, you guys seem like an energetic bunch. There's certainly a lot of talking out there. I hope you keep up the energy levels. It'll help me for sure. Okay, so the first thing I'm supposed to do is introduce myself. And I'll be completely honest, I hate introductions, all right, for a couple different reasons. So first of all, you know, I give a lot of scientific talks throughout the year, and they always start with an introduction where they, you know, talk about how esteemed someone is. Well, that was very nice of Sandra and everything. And they list your awards and achievements. And I think it's a real problem because the sum of a person is more than your achievements. You know, life is more than your career, and who you are is more than simply what you're good at. Hopefully that's something you guys already know, but hopefully it's something you won't forget. In the case of speaking to students, what I really worry about is coming off as prescriptive, right? If you do these things, if you behave this way, you can be just like me, right? I think that's like the worst advice you could ever get. The best advice I got in this regard was actually from a Nobel Prize-winning scientist who reshaped the entire field of organic chemistry in the 20th century. And what he said was this: why would you want to be like me? The world already has me, they've only got one you, all right? Just be yourself. So I have to give an introduction, but I don't want it to be an introduction where you feel inclined to live life the way I've lived, okay? But I would be remiss not to give you any advice based on my experience. So I'll take it with that caveat. I'll give you a couple pieces of information just to be instructive and to give you my kind of bland background. Okay, I grew up in Delaware, you know, the most boring of the 50 states. Middle class suburbs, nothing really remarkable. My mother's a high school teacher, she teaches French. My dad, like most dads actually in Delaware, worked for DuPont, which is the major industry in northern Delaware. So most of my friends' dads were also chemists at DuPont. They were the major employer. Now, the one thing that is kind of interesting is that my dad actually grew up in a small village in India. And his family, very fortunately, understood the importance of education, because if you wanted to escape the poverty of rural India, you needed to go to college. If you wanted a good job, you needed an employable skill. There was only one community college in the area, so all my 7 aunts and uncles and my dad all went to college and majored in chemistry, okay? And so when I was in high school, I figured that the last thing I wanted to do was also be a chemist, because clearly, having experienced all of this, I knew I wasn't good at it. I wasn't good at memorization. I'm still really not. That's why I have these notes. I never had a real flair for math. What I was really interested in actually was art and music. And I viewed science, especially chemistry, as diametrically opposed to art. I mean, clearly I'm not alone. If you look at how art programs and music programs across the US are being defunded, clearly people see it as useless. And I think nothing could be more foolish or short-sighted or ignorant to what science is. Right? I'm going to give you a feel for that today. Above all, what science requires is creativity. if we don't let you guys, the upcoming generation, be creative, we're going to kill science. And hopefully, as I'll show you today, the more creative you are, the more likely you're going to be successful at science. So anyway, I went to college at Penn State. I decided to be a pre-med major because, and I kid you not, I wanted to make a lot of money. All right, that's like the worst reason to do anything in this world. But then I took organic chemistry. And not everyone who's an organic chemist grew up playing with Legos, but a lot of them did. All right, if you like Legos, I don't know, do people still play with Legos? If you like Legos, if you like Minecraft, I guess that's the new Legos today, you're gonna love organic chemistry. It's not easy, it's actually pretty hard, but there's actually a lot of it that's like Legos. It's kind of like classical music composition, kind of like improvisation, and it's all hung on this visual framework for understanding the chemistry. I think that's a lot like Legos and Minecraft. So it appeals to visual thinkers, and particularly to artists, and in some ways also musicians. There'll be a little organic chemistry today, but not a whole lot, and I'm going to focus more basically on science. The first thing I think I ever did right in my education was to get into a research laboratory in college. So for those of you who are interested in going to college, for those of you who are interested in going on in your education into science, I highly urge you to try to get into a research lab as soon as possible. We're talking like freshman and sophomore year. You're going to have to try to balance it with your classes, but it was the best choice that I made, and it's something that higher education institutions like Scripps actually looks for in their applicants. So if you want to do science, you basically have to get a PhD. What that means is 4 years of college. I went to Penn State. 4 to 5 years of a PhD. I went here actually to the Scripps Research Institute. And then many people do 2 to 3 years of a postdoc. The nice thing about US science is that you're actually paid while you get your PhD and while you do a postdoc. So you're actually accruing debt through that education, which I think is really useful. Now, is there a secret to success in science? I think there is. Okay, and I'm going to give you 2. The first one I'm not going to elaborate too much on, but I think it's valuable because it's not something that I heard enough when I was growing up. And that is work really hard. There is no replacement for a good work ethic. I'm going to repeat that again. There's no replacement for a good work ethic. Accepting responsibility, putting entertainment as your last priority, and just being a mature, thoughtful person can really take you a long way. Okay, that's the only, I know I'm really lecturing to you here, okay? But that's the only kind of finger-waving advice I'm going to give you today. The second thing is I'm going to teach you some guidelines that I hope will be useful in general for not only understanding what science is, but doing science in general. And maybe it'll be applicable beyond just your education and your career. So what we'll be talking about today is essentially knowledge. Where's my pointer here, okay? Essentially knowledge and viewing science as an algorithm for understanding knowledge. Some people call this algorithm strong inference, okay? So what is knowledge? What do you get it? And if you think you have it, how do you really get it? There are many, many types of knowledge. Scientific knowledge is just one of them. The scientific algorithm is only one way of getting knowledge. So what are other ways of getting knowledge? Does anyone here have a favorite song? Anyone here have a favorite song? You want to raise your, are you willing to sing your favorite song? No volunteers. Suddenly those prizes look a little less appealing. I got one hander, red shirt, and Coca-Cola. No lyrics, okay. Well, okay, I won't make anyone sing. So, you don't necessarily have to recall and know your favorite song by using the scientific method. You're not running any experiments to know your favorite song. Okay, that's transmitted knowledge. Okay, you don't have to, it's not acquired via the scientific method. So, the scientific method is one way among many of attaining knowledge, but I think it's a pretty good one, all right? So, what I'm going to talk about today is how to acquire knowledge through the scientific method. So, here's an old picture. This is actually pretty hard to find. Anyone recognize any of these people in the picture? It's a little esoteric. Some mumbling. No. Oh, red shirt. No. Okay. Well, I was really hoping he had somebody. So this is a group of scholars from Oxford in the mid-20th century, 1940s and 50s or so. Okay. Some of them you might know actually. So this might be Charles Williams. The other one might be Owen Barfield. No one's usually heard of them unless you're really into literature. But the other one is J.R.R. Tolkien. Okay, here, and this is C.S. Lewis. Now I've got 3 little kids. Okay, so I'm big into children's literature, and you probably recognize them as the authors of *The Lion, the Witch, and the Wardrobe*, which many people have read. Okay. Oh, now you recognize them. Okay, and J.R.R. Tolkien wrote The Fellowship of the Ring and The Hobbit, okay? Both of them would be aghast at all of these commercial products like Happy Meals and Legos that grew out of their books. Okay, so what has sort of been lost, particularly given the movies and the toys and all of that, is the fact that these guys were serious literature scholars, okay? J.R.R. Tolkien studied Anglo-Saxon literature and Old English. He wrote a definitive translation of Beowulf, which is the oldest Old English long poem in existence, okay? He would have hated this kind of stuff. He was a very old, curmudgeonly, devout Roman Catholic guy. He really bemoaned, actually, commercialization and the industrialization of England as you saw it at the time, where factories were going up, fields were being destroyed. And if you can read that sort of as a subtext within all of his books, The Lord of the Rings. The other one is C.S. Lewis. Yes, he wrote The Lion, the Witch, and the Wardrobe, but he was one of the most celebrated literary critics of the 20th century. Okay, he's on the cover of Time. He wrote poetry, he wrote adult fiction, he wrote philosophy, he wrote theology. He was incredibly prolific, and one of his points of distinction was that he was considered one of the best debaters of his age. In fact, it's said that of the 100 debates that he engaged in, he only potentially lost one. One of the theories about the origins of his Narnia books is that they were actually a response to that lost debate, all right? And given their wild popularity, you might maybe reconsider whether he won or lost that debate at all. So Lewis was an incredible debater, and he had an incredibly penetrating mind, all right? And I'm going to tell you a little bit about how he came to have the kind of penetrating mind that he had, all right? So Lewis grew up in Belfast, Ireland, okay, here. And he grew up, he had a brother. His mother died when he was very young, and he was raised by his kind of eccentric father. And when he was about 14 or 15, the same age of some of you in the audience, he actually went to boarding school all the way down here in Surrey, just southwest of London, okay? So he was completely thrown out of his context, and he was told that he was going to study under this very nice, grandfatherly old headmaster who had taught his father actually when he was young, and he was going to have a wonderful experience. So he crossed the Isle of Man, took a train down to London, and then down to Surrey, and he was met by horse and carriage. Okay, this is the early 20th century. At the train station by the headmaster of the school, who is referred to here as Kirk. So he was very awkwardly sitting here in this train carriage, riding to the school from the train station, and he's trying to make conversation with someone who is, 60 years his senior. And so he said, and I'll read from his autobiography now. I said I was surprised at the scenery of Surrey. It was much wilder than I had expected. "Stop!" shouted Kirk, with a suddenness that made me jump. "What do you mean by wildness, and what grounds had you for not expecting it?" I replied, "I don't know what," still making conversation. As answer after answer was torn to shreds, it at last dawned on me that he really wanted to know. He was not making conversation, nor joking, nor snubbing me. He wanted to know. I was stung into attempting a real answer. A few passes sufficed to show that I had no clear idea responding to the word wildness, and that, insofar as I had any idea at all, wildness was a singularly inept word. "Do you not see, then," concluded Kirk, "that your remark was meaningless?" I prepared to sulk a little, assuming the subject had been dropped. Never was I more mistaken in my life. Having analyzed my terms, Kirk was proceeding to deal with my proposition as a whole. On what had I based my expectations about the flora and geology of Surrey? Was it maps, or photographs, or books? I could produce none. It had, heaven help me, never occurred to me that what I called my thoughts needed to be based on anything. Do you not see, then, that you had no right to have any opinion whatever on the subject? So this was C.S. Lewis's introduction to the next 3 to 4 years of his schooling, okay? And while some people, other students at the school, absolutely detested the method of this headmaster, Lewis described it to him as red meat and strong brew. He absolutely loved it, absolutely took to it, and it turned him into this wonderful debater. Okay, so what was the problem about what Lewis said? So what Lewis did was he expressed his opinion about Surrey that was based on a belief about southern England which did not line up with fact. And therefore you could say he had no true knowledge. Okay, so let's look at this in a visual way. You have the body of information, fact, okay? Whatever it is, physical world, geology, flora. You have another body of information, belief. These are your opinions. These are the things you think about. These are the propositions you have. Where they overlap is knowledge, okay? They don't necessarily have to overlap. You may believe things that are not true. There may be facts out there that either you don't believe or you don't know about, and therefore it can't be said that you have knowledge in that area. So belief in something that is not true is not knowledge, and truth that is not believed does not cease to be true, but cannot be called knowledge. Everybody got it? It's a little complicated, right? The scientific method is an algorithm for acquiring knowledge. In other words, it asks the question, how do you know what you know? How do you know that you know? Okay? Knowing something is kind of a complicated feat. So let's refresh what the scientific method is. And I'm going to put it in kind of just everyday terms, okay? You've probably in your science classes come across observation, hypothesis, experiment, conclusion. That's fine, and there's a lot of importance actually to knowing those terms, but it kind of divorces the scientific method from everyday life, okay? And you can use this in pretty much anything, okay? So in the scientific method, you have a question, you have a problem that you want to answer. You think about possible answers, you think of the property of those possible answers and how you might then observe them, okay? You observe, you make an experimental test, and then you can ask another question. So let's just start off with something simple, okay? And the question I have for you here is, what is the color of water? Sounds easy, doesn't it? Anybody? So here we have 4 pictures of water. We have clear water from the tap, kind of gray, murky water, and there's a large body of water. Here we have some water that looks green. Here's the deep blue sea, right? So we have some blue water. So what do you think? What's the color of water? We're on hand raised.
Water has no color. The color that we see is based on what it is reflected by.
Okay, so so what he said is water has no color. The color that we see is the color that it reflects, okay? And so you can see here's a gray sky, here's a gray body of water, here's some green, maybe it's, I don't know, some dirt in there or something, and then maybe there's a blue water reflecting the blue sky, so he says it's clear, it has no color, okay? Anybody else? The gentleman with the red shirt said that maybe there's some impurities in the water that are giving the water some color, okay? It's a good thing, I guess, that this is clear, right? Okay, so let's think that here are possible answers. No color, we got 2 votes for no color, gray, green, or blue. So let's think about the properties of the possible answer. So you guys, if you drink juice, right, you pour a glass of juice. If you pour a little bit, you look down into it, kind of let's say it's cranberry juice, kind of pale red. You pour a taller glass of juice, you look down into the glass, it's darker red. This is actually a chemical property called Beer's law, ironically. And it says that the deeper the water, the stronger the color. There's actually a guy named Beer, not because he was drinking beer. Okay, the deeper water is a stronger color, and so what we have to do is observe in a controlled environment, okay? So impurities in the water could cause it to look a different color, so we want to control it, make sure the water is pure. This is the importance of the scientific laboratory. The laboratory is a controlled environment where you're not influenced by external sources, okay? And then after doing the experimental test, we can draw a conclusion, and it allows us to ask another question, right? Is the water really pure? Is the environment really controlled? And we can go back and recycle these questions. Okay, so that allows us to ask another question. Okay, so one thing you can do is you can put the water in a spectrophotometer. That's really nice. Scientists like to use fancy gadgets like this. What it shows you is, where water absorbs and the spectrum is very, very sensitive. Okay, that's nice, but we can also use Beer's law, and we can make a really, really tall glass of water. So this is a 3-meter glass of water. That means it's about 10 feet tall, okay? And the interesting thing is if you look through it, it's actually blue. It's not colorless. It appears to be blue. So I just blow your minds or what? I don't know. This is pretty cool. So yeah, that's what I said. So why does the water appear to be blue? Okay. So there are a couple different possibilities, and we can go into more depth if you want. It could be electronic absorbance. It could be nuclear motions of the water. One possibility is that if it's nuclear motions, then the mass should affect the color. Interestingly, we can then change the nuclei rather than the electrons. And so if we swap the hydrogen for a deuterium, this adds 1 neutron to the atom of the hydrogen. Actually, this is not empty glass. This is filled with deuterated water. This is the kind of things that are used in nuclear reactors. Okay? And that's colorless. Okay? So it's pretty interesting. We can ask another question. One possibility is it's not a controlled environment, but let's go a little bit deeper. Okay? So the question is, do you now have knowledge about the color of water? Who says yes? See some hands. Who says no? See some more hands. The people that didn't raise their hands, I think, are the most right. Sorry, trick question. So, okay, the answer is maybe. The answer is maybe, right? So maybe only if you believe me. If you don't believe me, then it couldn't be said that you have knowledge, okay? And only if what I say is true. If what I say is not true, then it's not knowledge. Correct? So either my observations could be wrong, right? Or I could be incorrectly interpreting my observations, or I could be lying to you. I'm not lying to you, all right? But I guess it's a possibility. I could be lying about not lying. Or the source of my information is correct, okay? Maybe my internet search yielded the wrong Google image, all right? But anyway, my point is that to say that you know something is really complicated for you. How often do we walk through life saying, I know this, I know that, I have this or that opinion, all right? It's really complicated. So I'm going to tell you today about the scientific method and its ability to, in some sense, afford you confidence, not absolute assuredness, but confidence that you know that you know. Now, here's an interesting quote. Karl Popper, he's called the grandfather of science philosophy. And everyone who's in science or science philosophy, the history of science, knows Karl Popper, a major figure in the 20th century. He said there is no such thing as proof in science. What? How many headlines have we looked at in the newspaper that says, scientists prove this, scientists know that, okay? There's no such thing as proof in science, okay? Science instead is refutation by experience. So here's a quote from Karl Popper. There's no such thing as proof in science because some later explanation may be as good or better, so that science advances only by disproof. There's no point in making hypotheses that are not falsifiable. So that means you can't ask a question that you can't answer. Okay? There's no point in making hypotheses that are not falsifiable because such hypotheses do not say anything. It must be possible for an empirical scientific system to be refuted by experience. So if there's one nice synonym for science, I think it would be refutation by experience. I'm going to talk to you about another paper that I found very influential. You can pull this up. I think it's actually free online. If not, email me or one of the people that are in charge of this, and I can send you the paper. It's simply an editorial in the journal Science from 1960. It's actually nothing new, but it's John Platt, who's a mathematician and sort of interfaced also with biology at the time. And he wrote an editorial calling people back to remember the precepts of the scientific method. Because in some ways, the more advanced you get in science, the more you forget some of the early things that you guys are learning right now, and it's really valuable to think about them. John Platt comments on this idea from Karl Popper that science is refutation, science is disproof, okay? And what he says is that the difficulty is that disproof is a hard doctrine. The reason why, in some ways, the more advanced you get in your scientific career, the reason why you forget about these things is that you know a lot of other scientists, you don't always agree with them, right? And you don't want to lose their friendship, but what John Platt says is, if you have a hypothesis, and I have another hypothesis, evidently one of them must be eliminated. The scientist seems to have no choice but to be either stupid or a jerk. All right, I'm paraphrasing a little bit. Perhaps this is why so many scientists tend to resist the strong analytical approach and why so many great scientists are such jerks. He says, fortunately, this difficulty can be removed by the use of a second great intellectual invention, the method of multiple hypotheses. Okay? So let's unpack that a little bit. All right, so what does he mean? Well, his point is this. At the heart of science is exclusion of competing hypotheses. So you have an idea, I have an idea, we have to figure out which one is right. All right? A lot of times people view science something more like this. I have an idea, I prove it, and guess what? I'm right. feels good, right? We want to be right. We want our ideas to be novel and change the world. And I think that's a good thing. But this is not how science works, right? Instead, you're excluding hypotheses. You're not proving your ideas, you're disproving competing ideas. That's why there's no such thing as proof in science. So instead, science works not like this above sequence, but rather like this. You have ideas A, B, C, D, and E, and you eliminate some of them one at a time, or in concert, and find out that answer B is right. This is really important, actually, and maybe actually increasing in importance. So what John Platt's essentially saying is that we all have a vested interest in our own work and our own insights in being right, okay? That's just a fact of humanity. If you want to make sure that you're right, you've got to come up with your own competing hypotheses, not your own pet idea, but rather multiple ideas to actually get you at the truth, all right? There are multiple studies that suggest that, at least in the US, narcissistic personality disorder is increasing. There's actually studies that have been done right here in San Diego by Professor Twenge at I think San Diego State University. Okay. It's really important, and I think it underscores I think it supports this idea that multiple hypotheses are going to lead you to the truth faster than if you have one idea. Now, everyone wants to say, okay, narcissistic personality disorder, that is, a focus on the self is increasing, kids these days, all right? Don't forget that John Platt wrote his essay in the 1960s. Wait, scientists have been doing this a lot longer than you have, that is, being narcissistic, all right? All right, so let me give you an example of what this looks like from a chemistry perspective. So I'm going to ask, what's the bond order of benzene? This is kind of a complicated question, but let me unpack it for you, okay? So benzene is this molecule. It's a pretty fascinating one. This kind of structure is embedded in a lot of the drugs and medicines that people are taking now, okay, the good drugs, medicine. So it was actually a big question in the 19th century, because in the 19th century, street lights were not based on fluorescent light bulbs. I think the ones we have usually out here are sodium vapor light bulbs. Instead, they were based on burning natural gas, or in fact, in some cases, ethylene, which is a product of the ripening of fruit, okay? And, or coal. And what they found is that when they burned these natural gas sources, they ended up with this residue, okay, that contained molecules, including benzene and things that looked a lot like benzene, all right? And of course, that gunked up all the street lights. So they had to find a way to reduce this, and to understand how to reduce it, they had to understand the structure of benzene. It was a huge question that consumed dozens, if not hundreds, of scientists in the 19th century. So one of the questions they had was that, what does it actually look like? Okay, molecules are so small that you can't just, take a microscope and look at them. Today, maybe with some very big molecules, you have very, very specialized microscopes, but this was 200 years ago. All right, and the question they had was that, is benzene a mixture of these two things? Okay, this is kind of a simplification, so for people that actually know some organic chemistry, forgive me, this is simplification, okay? Is it a mixture of two compounds where one has, double bonds at these two positions, these two positions, these two, so you have 2 lines here, 2 lines here, 2 lines here, or does it have 2 lines here, 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 and you have a mixture in this liquid benzene, okay? Or is it actually not a mixture in the liquid, but rather is it some kind of hybrid of these two? So is actually none of these correct, but rather instead of drawing the 2 lines here alternating or alternating here, instead you could draw it as a circle, all right? So what do you have to do? Well, ask the question, what are the alternatives? And then what you have to think about is you have to use your imagination, all right? And this is also true of the example I gave with water, all right? At the heart of all of science is using your imagination. Thinking about, if something is this way, what are the consequences? What are the other possibilities? What are all the hypotheses, right? So multiple hypotheses requires you to be incredibly creative. You can't just have a great idea. You have to have like 10 great ideas and decide which one is right. All right, here I'm just going to give you 2. So if you have alternating double and single bonds, then you can draw compounds. You can draw, these are 5 different molecules, okay, all of which are different, all right? So X and Y can be around, this is a chemical bond. You can draw it as 2 lines. Or X and Y can be on this side where it's around 1 line. Or they can be apart from each other like this, or apart from each other like this, or across the ring from one another like this. So there are 5 possibilities. On the other hand, if you have a structure that's more like this, where there's a circle in the middle, okay, there can only be 3, right? This is one of the reasons why organic chemistry is so appealing. I don't really have to tell you anything about London dispersion forces, about the ideal gas law, okay? You can just see that these things are different. And you can just see that these things are different, but there are no other ways to come up with different compounds with X and Y attached to this, hexagon. Okay, and then what you can do is figure out an experiment to probe, okay, are there only 5 derivatives possible, or are there only 3 derivatives possible? Are there only 5 different molecules or 3 different molecules? Okay, and you can run an experiment to test that, and it turns out that there are only 3. Okay, and therefore you assume, okay, so the structure of benzene then is better represented by something with a circle rather than these alternating double lines. Okay, so the question then you ask is, are you sure? Are there other alternatives to explain the fact that you only see 3 disubstituted benzene and not 5 disubstituted benzenes? All right, and as Karl Popper points out, there's no such thing as proof. Even now, people could come up with a better explanation theoretically for the structure of benzene. All right? One alternative way to explain this is not that there are only 3 derivatives that you actually observe, but rather that the experiment you did only led to 3. So you got to do the experiment a different way. For instance, if you take a benzene derivative called anisole, this is actually very related to the vanilla that you find in all the artificial vanilla-containing products that you eat. Okay, so if you buy something that has artificial vanilla in it, it's actually related to benzene. It's kind of interesting. So if you chlorinate this, if you put X and Y on this benzene ring, it actually gives you only 2 derivatives. Now that's kind of weird, right? So you've got to reconcile all these different possibilities for explaining physical reality. What really slows you up in science is either not having the willingness to generate multiple hypotheses because you want your idea to be right, not having the knowledge. I mean, there is some schooling involved, right? But especially not having the creativity. All right, science is all about seeing the same thing that everyone else saw and thinking about it in a new way and coming up with a new hypothesis that better fits the data. So let's think about a question that's less chemistry. Okay, first let me show you basically what this would look like. Okay, so you have some kind of question you want to answer. You think about what are all the different possible answers to that question. And that requires using your imagination. Okay? So we thought about what's the color of water. We saw all this color of water around us in the world. We think about, okay, which one is actually the color of water? And so you think about this question. You come up with, let's say, 5 different answers. Okay? You think about the consequences of that answer through an experiment. Okay? So answer 1 will give you an experiment. Let's say through an experiment would be outcome A. Answer 2 would give you B. Answer 3, A. 4, B. 5, C. Well, you can see here that different answers can actually give you the same experimental outcome, right? So what would be a good, well, maybe we'll come to an example of that. You run the experiment, okay, and you find out what the outcome is. All right, let's say it's outcome B. Okay, well, that's fine. That eliminates now, let's see, what does it eliminate? Answer 1, answer 3, and answer 5. But if outcome B relates to answers 2 and 4, then you haven't actually differentiated between the 2. And so you have to come up with another experiment to differentiate outcome B. And so these are different answers with then, through another experiment, different outcomes. You find out which one is right. Since answer 2 leads to outcome D, then running the experiment gives you outcome D. Then you eliminate then answer 4. All right? So through these exclusionary logic trees, you've eliminated all your hypotheses to come up with what the right answer is. Now, is this the right answer? Well, that depends on your creativity to generate multiple hypotheses. So let's look at a concrete example. All right. So Santa Ana winds. We face them all the time. All right. I hate them. I'm from the East Coast. I'm not used to this dry weather. Okay. It feels very unnatural. And so I was really curious about, what are these Santa Ana winds and why are they hot? So just to refresh, what are Santa Ana winds? Anyone want to raise their hand? Oh, guy in the front. He says they're winds from the desert. Okay. And what do they generally do? What do they generally do around here or wherever you live? Someone in the front. What's that? They make your skin dry. Yeah, that's true. I've experienced that. So So, and why do they make your skin dry? Because it's hot. Because it's hot and dry. Okay, all right. So they're hot, dry winds. Okay, they blow through here. The temperature goes from 60 degrees to 90 degrees in one day. They're really irritating. Okay, so why are they hot? Why are they hot? So let's think about that. Okay, and to come up with different explanations, all you have to do is use your imagination. All right, and so listen. I'm not an environmental scientist. I'm not a meteorologist. Okay, I'm kind of just making this up as I go, but I think it's a good illustration. So I just use my imagination. So what are all the possible reasons for why Santa Ana winds are hot? Well, rather than solicit answers from you, I'm just going to give you my ideas, because then you can make fun of me. Okay, so one reason is that they're from the desert. So they're hot because they're from the desert. Desert's hot, wind blows here, makes here hot, okay? Okay, that's a possibility. They're also coming from the east. Maybe there's something about coming from the east that makes them hot and dry. They're coming over the mountains or from the mountains. So maybe it's not the desert, maybe it's the mountains that makes them hot. They're coming towards the ocean. Maybe it's the fact that it's going through the ocean that makes them hot. And the winds are fast, okay? The wind speed picks up. So maybe it's something about the wind speed that makes it hot around here and makes it dry. Okay, so what can you do to determine the basis for this? Well, one thing you can do is you can just look at other places in the world that experience these kinds of meteorological events, okay? So you compare either other regions, or you can look at this area at different times and figure out, okay, well, maybe it's that the winds are coming in, when do they come? Late summer. Maybe it's because it's late summer that they're hot. Okay, so what would be the consequences then of this comparison? So one consequence is that, okay, if you have a hot desert, that brings hot winds. That would mean that a cold desert would bring cold winds. So if the desert's cold and these Santa Ana winds come, then we should expect a cold climate. Are all east winds hot winds? Well, if the wind's coming from the east, does that mean it's going to be hot here? I don't know. They're descending winds. They're coming from the mountain. They have to come over the mountains. So if we look at another region where they have these kinds of events, but there are no mountains there, is it still hot? They're coming to the ocean. We can't get rid of the ocean. But if we can look at another region where there's not an ocean, but everything else is identical, and they have these kinds of events, what do we see? Are the winds always fast? Can they be slow winds? Can slow winds make it hot? Okay? So then all we have to do is basically compare other regions, and that will be essentially our experiment. Okay? Find the phenomenon elsewhere, or look at the phenomenon at different times under different conditions here. So I could do that. That would take a really long time. I thought I just Googled it. Okay? And it turns out, and I think this is really interesting because it actually involves chemistry. Would you believe it? So the phenomenon exists actually when you have high pressure over the desert. Actually, it can be cold in the desert, okay? And the pressure builds from so you have a high pressure area which flows into the low pressure area, in this case on the coast. And when it does that, it has to funnel through the mountain passes that lead from the desert over the mountains and down towards the coast, all right? When it does that, it acts just like a refrigerator in reverse, essentially, okay? So in a refrigerator, you pass a gas, okay, through a small tube and it expands, okay? And when it expands, it gets everything really cold, all right? And the heat transfers. You see those coils in the back of your refrigerator? They're hot, right? So it expels the heat out the back. Okay, this is called adiabatic heating and cooling. It's a fancy word. Basically, it just means that it's heating and cooling without a temperature, else is complicated. Okay, it's adiabatic heating and cooling. So let's get you into the thermodynamics and stuff. Okay, but anyway, it acts just like a refrigerator. So what happens, the basis for the Santa Ana winds are that you take this high pressure system and you're shoving it into the valleys between the mountain passes that compresses it. And when it compresses it, the air gets really hot. And when the air gets really hot, it heats up our climate. Pretty cool, okay? Now, I don't know how this was determined, but one way you could do it is simply by excluding these different hypotheses by looking at different regions, okay? So I'm going to humor myself here and just talk about some of the things that we work on so you can see how we use this idea of multiple hypotheses and exclusion through experimentation. So we're a chemistry lab, all right? But we work in topics in biology. And one of the topics we work on is malaria, all right? Malaria is a major health concern, okay? It's not something we have to necessarily worry about here because of the climate, all right? But it's a big problem, especially in low-income countries that lie along the equator where mosquitoes are a really big problem. All right? And so if you look at low-income countries, malaria is a major health scourge. It's the number 5 top killer of people. So it's about 5% of the total number of deaths. And the vast majority, actually, of the people who die are children. And in Africa, 1 child dies from malaria every minute. Okay? And that's because their immune systems are not strong enough to repel the parasite. Okay? Malaria is a parasitic infection. The countries in the equatorial region, so generally around here, are affected by malaria. What I'm showing you here are not just countries affected by malaria, but countries where there's resistance to the current medicines that are used, the current drugs. Okay? So chloroquine is a very famous drug. It was based actually on a molecule found in the bark of a cinchona tree from South America. Okay? It was one of the original antimalarial drugs. And because it was used so early, now through evolution, the parasites have evolved resistance against this drug. And so if you're in this country and you get malaria, there's a good chance if you take chloroquine, it's not going to cure your malaria. Then you have to find a new medicine to take. The areas that are circled have resistance to not just chloroquine, but other antimalarials. Okay, so you have multiple drug-resistant strains of malaria. So now you have to go through various regimens of treatment from your doctor, which of course have side effects, to try to cure yourself of malaria if chloroquine and these other antimalarials don't work. The really scary thing is that just as of 2009, which is pretty recently in the grand scheme of things, they found hotspots of malaria resistance to artemisinin. Now, artemisinin is kind of like the wonder drug for malaria. People thought it would some people thought it would eradicate malaria across the planet, okay? But all of a sudden we started seeing parasites to which artemisinin was not effective. It wouldn't kill the parasites. And this is very scary. And in fact, the World Health Organization has now put out a call for people to come up with solutions to either develop new drugs to replace artemisinin or solve this resistance problem. This could turn into a real epidemic in malaria-stricken countries. So our lab is more interested in chemistry. Here's the structure of a molecule. It just looks beautiful, doesn't it? I mean, it's let's just bask in the molecule. I mean, it's an incredible structure. Now, interestingly, this molecule is found in a sponge, okay? So all of us are made up of molecules. I don't know if you've done cellular biology yet, but all of our cells are made up of phospholipid membranes. These are just greasy molecules, kind of like soap, okay? We have DNA. DNA are just huge molecules, okay? But we also have really small molecules like this. So hormones. Hormones are molecules, all right? What's a good example? There are molecules secreted by your thyroid that control calcium, efflux and influx. So all of our bodies are governed by molecules. All of sponges' bodies are governed by molecules. Sponges are actually incredible producers of really interesting molecules, many of which are actually poisonous. But why are they poisonous? What's the difference between a person and a sponge? That's a big question. Okay, a lot of differences, right? Unless you're SpongeBob, I guess. He's pretty similar. So one of the big differences is the sponges can't run away, all right? There are no, if there's a lion, we run away from the lion. There's no lions on the bottom of the ocean, but there are predators, right, that eat sponges. And if you're a sponge, you can't run away. But you can make some nasty molecule that either kills a predator or makes your body taste really bad, okay? And so these sponges actually have produced a lot of molecules that are poison, and because they produce these poisons, they're actually really useful. So there are a lot of anti-cancer drugs that have come from things like sponges that are in clinical trials right now, and that's because you want to kill the cancer in your body. And the sponges can produce molecules that can do that in a general way. One of the molecules it produces is called calilanol A. This is calilanol A. One of the things it's good for, it turns out, is actually preventing what's called biofouling. All right. So if you have anything that you stick into the ocean for a long time, whether it's this current meter, this measures how the ocean currents, or whether it's like a ship, all right, you're going to start first growing bacteria on it. They form a thin film, and then other things like mollusks build up. If you're shipping things across the Pacific Ocean, this creates a lot of drag, all right? That burns a lot of fuel, pumps a lot of carbon into the atmosphere, consumes a lot of money, all right? So the shipping industry knows that biofouling is a huge problem. Sponges have known that for a long time. If you're a sponge, you don't want the color covered with mollusks, all right? And so one of the reasons that people think this sponge secretes this molecule is to prevent this biofouling. It doesn't want things to grow on it, especially bacteria that can kill it. What's really interesting is that this molecule not only prevents biofouling, but it also kills malaria. okay, the parasites that cause malaria. That's a little bit weird. There's no malaria at the bottom of the ocean. Mosquitoes don't do so well in the ocean, okay? So what our lab does is we're involved in organic chemistry. We like putting molecules together. We're good at putting molecules together. And we use that ability to look at the structure of molecules to then study the biology, okay? So we want to know, how can we make this efficiently? Okay, one of the big problems with molecules derived from sponges is that if you want to treat humans, you gotta scrape all these sponges off the bottom of the ocean. Sometimes they make very little of the molecule. In some cases, to treat one person would render the entire sponge community extinct because they make so little of it. Okay, it's just a material problem. The other reason we want to make this molecule is to try to determine, how is this killing the malaria parasite when it's supposed to be just preventing mollusks from attaching to a surface, okay? Interestingly, for this molecule, there is one hypothesis put forth in the literature for how it's acting, but there was only one hypothesis put forth, not many. And there wasn't any experiment to differentiate between many hypotheses. There were a lot of experiments just to prove that the hypothesis was right. Does that sound like a good idea? No, 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 okay? Exclusion of competing hypotheses is very important, okay? So we came up with some interesting chemistry that for a long time people have thought impossible. I'm not going to bore you with the details, okay? It had been unsolved for 117 years of organic chemistry. That just makes me kind of feel good. But the part of the molecule we're able to make very efficiently is actually found in a lot of different kinds of drugs, all right? And it is found in the molecule that we wanted to make to treat malaria. If you wind up taking OChem, this will probably be on an exam, actually. But we showed that actually a lot of the questions they're asking on exams now, because of our work, can be rendered false, all right? So you can use that on your test if it helps you. All right. Anyway, so we made this molecule. Now, previously, molecules like this had been made in an average of 30 steps. So what does that mean? So chemistry, depending on who you talk to, is a lot like cooking, all right? You're mixing stuff together, you're filtering stuff, you're stirring stuff, you're heating stuff up to cook it, all right? So imagine if you're cooking a recipe that required, basically to make 30 different things. Not just making a cake, but then taking that cake and turning it into something else, and then taking that and turning it into something else, and doing that 30 times, okay? Not only is that incredibly laborious, but it actually takes a lot of money to do that. We can make this molecule now just in 7 steps, so we've cut this by almost a quarter. That saves a lot of money. You can imagine if this is kind of like making a cheese souffle, okay? I Googled that. That's apparently like the most complicated thing to make in the kitchen, all right? This is just a piece of cake. Okay. So, all right. So the question is, man, that's what I get applause for? Holy smokes. All right. So the question is, what we wanted to ask now, okay, is how does this reaction work? How does this amazing reaction we discovered work, okay? And so, we thought about how it might work. We used our imagination, we used our knowledge of chemistry, and we used our willingness to explore multiple possibilities, okay? And we came up with 2 options called ion pair attack or concerted attack. It's actually pretty easy to understand, all right? Especially if you played with Legos. I don't know if this is available in Minecraft, but in Legos it's pretty understand, all right? So if you have 2 Lego pieces stuck together, you want to replace one of the pieces, all right? You can either take it off and put another one on, or if you've got one of those pieces with a hole in it and you put a little peg in that hole, you can just take another peg and push one out push it out as the other one's going in, okay? So in some chemistry, it's called associative versus dissociative. In this case, it's called SN1 versus SN2. But it's really easy to understand if you played with Legos, okay? So I won't dwell on this, but basically, we've been able to design an experiment where we think could answer these 2 questions. It's based on what's called a kinetic isotope effect experiment. The take-home message is that we don't know the answer yet because Congress delayed our funding, so we're only now, if you guys have heard of the sequester, all right, have you heard of the sequester? That's so 2013, but basically, I mean, that prevented money from coming in that would fund this experiment to answer this question, all right? Okay, anyway, so that briefly describes how we put the molecule together, but I'm going to dwell a little bit more on another point, and that is, what we wanted to answer is, how is this molecule killing the parasite, all right? And at this point, there was only one hypothesis put forth. So let me just give you a crash course in malaria biology. All right, you can actually you might actually learn this in one of your high school biology classes that you take. It's pretty straightforward. So malaria is transmitted by a mosquito vector, an Anopheles mosquito. All right. So what that means is that the parasite basically hitches a free ride in the mosquito's salivary glands. All right. And that means when the mosquito bites you and takes a blood meal, okay, they suck your blood. Sometimes a little backwash, right? So the parasite saliva gets into your blood. All right. And when the saliva gets into your blood, the parasite is transmitted. All right. Now, a lot of people know about malaria from the perspective of the fact that it infects your erythrocytes, your red blood cells, okay? But it doesn't do that first. Actually, the first thing that happens is the parasite gets into your hepatocytes. These are the cells of your liver, okay? It has to grow there, it has to mature, get a driver's license, that sort of thing. And when it matures a little bit, then it bursts out of your hepatocytes and gets into your erythrocytes, your red blood cells. And this is where some really interesting metabolism happens, okay? So, just like you and me, the parasite needs some protein to survive, okay? If you're a vegetarian, you got to get it from beans or tofu or something, okay? If you're meat eating, you usually get it from, cheeseburgers or whatever. Not the healthiest menu, I guess. But anyway, everything needs some protein to survive. And what the parasite does is it digests your hemoglobin. Your hemoglobin is a protein, okay, that carries oxygen through your red blood cells. So when you breathe, you take in oxygen, the oxygen binds the hemoglobin in your red blood cells, your blood circulates it all over your body, and that causes respiration to occur in your cells. The parasite uses the erythrocyte as its source of protein. So those are the cheeseburgers that it eats. Now when it eats it, it has to get rid of heme. All right? Heme is a small molecule, kind of shaped like a dinner plate, all right, that's found embedded in the hemoglobin molecule. There are 4 of these on a hemoglobin protein. And it's actually toxic. If it weren't in the hemoglobin protein, it would kill your cells, all right? And so the parasite has to deal with this. When it digests your hemoglobin and it releases the heme, it's basically going to kill itself unless it does something with the heme. And what the parasite does is it takes this flat dinner plate molecule and stacks another one on top of it, stacks another one, and another one, and another one, and another one, and basically crystallizes it, all right? Kind of like a snowflake, right? It's all these molecules stuck together. And when it's stuck together, actually it's not toxic anymore, and it's just excreted from the cell, okay? So the hypothesis in the literature is that these molecules that we were making were killing parasites because it was preventing this heme from stacking, okay? I could bore you with the details. Basically, these molecules are thought to bind in an iron and then prevent the stacking from occurring. And what they did basically is the authors had 5 different experiments where they showed that these molecules can bind to the heme that's released, and therefore maybe it's then poisoning the parasites, okay? But they never ran an experiment that could exclude this hypothesis. They were trying to prove it. They were not trying to disprove it, right? So what we thought is maybe we can try to disprove it. Is there an experiment you can run to disprove the hypothesis? And there is. Now remember what I said, the parasite doesn't first go into your red blood cells, first it goes into your liver, right? So if you can test this molecule in the liver and show that it's still active, then it disproves the idea that it's targeting heme because there's no heme metabolism in the liver. There's not a whole lot of hemoglobin in your liver cells, okay? So what you can do is you can test the liver stage schism, okay? This is before the parasite has earned its driver's license and gotten out of the liver. And if you test it and it's efficacious, then it has nothing to do with this. Now, this is actually a really difficult assay to do. You have to outsource your mosquitoes to NYU in New York City, and they have to infect, first they have to grow up the mosquitoes, the Anopheles mosquitoes. And you know how they do this? So this is interesting. So mosquitoes, they don't like dead blood, okay? They want live blood, okay? And so if you're a technician, you're working in the mosquito lab, you take a cup of mosquitoes and you stick them on your arm. And you let them drink your blood, okay? Now this is before they've been infected with malaria, obviously, because that would be a problem, all right? Okay, but you have to grow these mosquitoes up, basically sucking on live human hosts. And then you have to infect the mosquitoes with malaria, okay? And then you have to sedate the mosquitoes, and you have to dissect out their salivary glands, right? How small is that? And then you have to take the salivary glands, and you have to get the parasites out of the salivary glands, and you have to put the parasites into human liver cells, and then you have to test your molecule on those cells. We don't do that, okay? But one of the labs we work with at UCSD does, all right? And I'm very happy that they do that. And working with them, we determined actually that our molecule does inhibit this liver stage parasite. And what that does is it completely overturns this decade-old hypothesis that it's inhibition of heme detoxification that's at the heart of these molecules. So now the hunt really is on to figure out what the real target is. How are these killing parasites? And if we can figure that out, potentially it could lead to a new drug for treating malaria. Okay? So all I'm going to leave you with now, to the take-home message is that science involves disproof. Okay? It's refutation by experience. Science is disproof of multiple hypotheses, so you don't have your own pet idea that you want to approve, okay? And you've got to come up with your own multiple hypotheses to do this, okay? Now, one of the best things about science is working with all the people in it, all right? This is an old picture of my lab, but here we are enjoying one day out of the lab. That's all we take off during the year. On the beach is Coronado, and the other great thing is that you get to work with people from a lot of different countries, all right? Science is worldwide, so we have people from, hope I can recognize all my flags here, so Spain, China, Italy, the United States, Japan, Italy, the United States, and just a chemist. I'm not, a politician. Anyway, China, Canada. We're leaving you on a good note here. It's been a long time since I took geography. So anyway, all of these different countries. And with that, I want to thank you very much for your interaction, for your attendance. I'd be happy to take questions.